Hi, I'm Joy Lim from Duke NUS Medical School. I'm going to be describing our recent paper entitled SARS-CoV-2 Breakthrough Infection in Vaccinated Individual Induces Virus-Specific Nasal-Resident CD8 and CD4 T cells of broad specificity. Tissue-resident T cells are localized T cells found in different anatomical locations and are important for local immunity and recall responses. Since SARS-CoV-2 infect the airway, we are interested in the nasal resident T cells as they might provide an immediate layer of protections against SARS-CoV-2. Thus, our objective of this study was to evaluate the impact of parenteral vaccination on the presence and functionality of the nasal resident T cells in vaccinated individuals with or without breakthrough infection. We deployed a simple sampling method using nasal swabs to locally isolate nasal T cells. We performed the localized sampling in naive vaccinees and in convalescent vaccinees. To determine the SARS-CoV-2 specificity of nasal cells, we performed an interferon gamma ELISpot assay. We stimulated the nasal cells overnight with different SARS-CoV-2 peptide pools, including membrane, nucleoprotein spike, and non-structural protein NSP12, which is the RNA-dependent RNA polymerase of the virus. So here are the results. Each stacked bars represent the T-cell response from individual donors, and the different colored bars are their responses against different SARS-CoV-2 antigen. In 16 naive vaccinees, we did not detect any SARS-CoV-2 specific nasal T cells, and this is the complete opposite for the convalescent vaccinees. In almost all of them, we were able to detect nasal T cells targeting different SARS-CoV-2 antigens. In fact, majority of these subjects responded to 2 to 4 different peptide pools. Thus, broadly specific SARS-CoV-2 nasal T cells are induced in vaccinated donors only after infection. Next, to further characterize the phenotype and function of the antigen-specific nasal T cells, we performed an activation-induced marker assay. We detected both CD8 and CD4 antigen-specific T cells in 6 convalescent vaccinees. Interestingly, majority of the CD8 antigen-specific T cells are CD69 and CD103 positive. Thus, they are made up of mainly tissue-resident CD8 T cells. But that is not the case for CD4 antigen-specific T cells. Then, we cross-sectionally and longitudinally looked at the persistence of nasal T cells. We found that antigen-specific nasal T cells remains at detectable levels stably for at least 3 months. The longitudinal analysis also revealed that different antigen-specific nasal T cells wanes at different rates. Lastly, we compared the T cell responses from the nasal cavity and the blood. On the right are the responses from the peripheral blood, and on the left are the responses from nasal cavity of the same subject. We saw that dominant responses from the blood were almost always targeted against spike. However, the dominant response detected in the nasal cavity came from MP in grey, NSP12 in blue, and spike in red. Therefore, the hierarchy of SARS-CoV-2 T-cell responses were different in the blood and nasal mucosa. In summary, SARS-CoV-2 infection induces localized SARS-CoV-2 specific CD4 and CD8 T-cell responses. Vaccine-induced spike-specific T-cells do not suppress induction of T-cells specific for other SARS-CoV-2 proteins. Hierarchy of SARS-CoV-2 T-cell responses were different in blood and nasal mucosa, and importantly, nasal resident T-cells might explain why hybrid immunity is more protective than 3 doses of spike mRNA vaccine.